please welcome Chancellor Coleslaw.
Good afternoon, everybody, and welcome. I thought he will never introduce me the way he did, but I'm glad he did. But really, the most important thing today is introducing our esteemed guest, Dr. Venkatraman, also known as Venki Ramakrishnan. It's indeed a great honor, Venki, to welcome you back to, if I may say so, your alma mater or your second alma mater. Venki is an alum of this place. He did his postdoc here. And I think he will share with you at some point that this is the place that changed him or changed his career in a very good way. So during the afternoon when I was talking to him, he said he had two choices at some point, to go to UCLA or to come to UC San Diego. And he looked at both places and hands down, he knew it was going to be UC San Diego. At least that's how I remember it. You know, being the chancellor, I do have a jaded view of life, but that's how I remember it, and maybe you can correct me. But let me just say a few words about Venki. He is an amazingly accomplished person. He's done a lot in his life, and I think it's just fair for me to read his bio verbatim. So Venki is a senior scientist at the MRC Laboratory of Molecular Biology in Cambridge, England, and the president of the Royal Society. This is one of the most prestigious societies in the world, not just in the UK, and it is way older than our national academies. He's also currently a fellow at Trinity College. He earned his Bachelor of Science in Physics at Baroda University in India in 1971. Most of you would not have heard about it. And during a time when India's best and brightest were seeking opportunities not available in their home country, Venki enrolled at Ohio University at the age of 19. And he was awarded his PhD in 1976. So in 71, he was 19. In 76, he was 24, and he had a PhD from Ohio U. And the way the conversation went this afternoon, he was looking to do something different. And this, I believe, was in physics, right? The PhD was in physics. And he knew at that time that biology was where the action was going to be. So he wanted to do something in biology. Okay, so following this achievement, he switched his career path to life sciences, beginning as a biology graduate student at UC San Diego. I'd like to think that UC San Diego's non-traditional pioneering approach to all we do was uniquely suited to fostering Venki's non-traditional approach to his training as a physicist turned biologist. And he left La Jolla after 2 years for a postdoctoral fellowship in the Department of Chemistry at Yale, and went on to advance his groundbreaking work on the ribosome at Brookhaven National Laboratory and the University of Utah. And he received the Nobel Prize in Chemistry for his work on ribosome structure and function in 2009, and he is a Knight of the British Empire, an elected member of the National Academy of Sciences and the Indian National Academy and the Indian National Science Academy, and the recipient of countless awards and honors. He is indeed a beacon of inspiration, not just to all of us out here, but to a broader community that's doing research in this area across the world. And so we are indeed very honored to have him back. He's back here only for one day, but I did offer him to come and spend a little bit more time with us next time. So with that said, welcome, sir. The podium is yours. Dr. Venki Ramakrishnan.
So thank you very much. It's a real pleasure to be back in La Jolla, a place which really transformed my career and my subsequent life. So I'm here to talk today about a scientific memoir which I've called Gene Machine. And it's available in 2 different forms, as you can see. It was published last fall. So there are 4 themes in the book. The first is the central character in the book is this amazing molecular machine that's one of the oldest molecules of life. And it's the molecule that reads the code of life and really helps to build life, build organisms. And that includes everything from bacteria to all of us. And a second thing is what it was like as an outsider. I didn't go to, until I came to UCSD, I didn't go to really top-notch institutions, certainly not top-notch research institutions. And, I was an immigrant to the US at the age of 19, as you heard. Then I immigrated later in life to England. And so what was it like to be an outsider who found himself in the middle of a high-stakes race for which everyone knew was a really fundamental problem in biology? And the other people in the race were all much better established groups, much better connected as well. A third thing is scientists often, when they talk about their work, they talk about it in highly sanitized terms. Everything in hindsight was very logical. Everybody was doing the right and noble thing and so on. The reality is it doesn't work that way, because science as an enterprise is very pure. There's a scientific method and there's hypothesis and evidence and so on. But scientists are a different story altogether. Okay, scientists are humans, and so they have personalities and ambitions and egos and rivalries and all sorts of things. So, and you throw prizes into the mix. That's like adding gasoline to a fire. Okay, and then there are some lessons which are too late for me now, but maybe if there are young people in the audience, they might, get something from it. So to get back to the problem, we've all heard of genes. You talk to the average person and you mention genes, they all nod their head. You mention DNA, they nod their head too. They think they know what it means. And what they really think is, genes are things that we have that sort of make us what we are. You can have good genes or bad genes. Somebody's a mass murderer, they have bad genes. Somebody's a genius, they have good genes, etc. But the reality is, if you probe people a little further and ask, well, what exactly is a gene, they won't be able to tell you, okay? And when genes reside on a molecule called DNA, and when people started sequencing all of our DNA, you could get an estimate for how many genes we had. And this is an old slide. I really need to correct it, because here it shows humans have a 35 to 45,000. Actually, we only have about 25,000 genes. And that's about the same number as a weed, Arabidopsis thaliana, or, only slightly more than a worm, okay? So anytime we think we're so much superior to other life forms, we're kind of kidding ourselves, okay? It's because we're fooled by our intelligence. We think intelligence is what's important about life. Life doesn't care about intelligence. Life only cares about survival. And we evolved intelligence, in Africa to survive, and that's how we, drove our own evolution. Worms, survive in different ways, and bacteria, not intelligent at all, but they survive just fine. So if you ask what genes are, what are these 25,000 genes? Well, each gene is a kind of a recipe on how to make a protein, okay? And so then you ask, well, what are proteins and why are they so important? Well, proteins carry out nearly all of the functions of life. And so what are they? They're long chains of building blocks, chemical building blocks called amino acids. And if you were to represent them as a chemical diagram, this is what a protein would look like. It's a long chain, and you can see these different groups sticking out, and the different groups have different shapes and different chemical properties. And there are about 20 of these in nature. So every protein is a different combination of these 20 building blocks. They can be different sizes. Some of them are shorter, some of them are longer, and the order of the amino acids is different in each protein, okay? And you can represent it in a very schematic way. So since there are 20 amino acids, you can represent each amino acid by letters, since we have 26 letters, so there's more than enough to go around. And you could just represent it as a string of letters, and that's what, conceptually what a protein is. Now, imagine if I told you I took a strip of paper, or several strips of paper, and I wrote down different sentences on these strips of paper. And depending on what I wrote down, each strip of paper magically folded up into a completely different shape, okay? That is exactly what happens with a protein. So the information on how this chain folds up into a three-dimensional shape is contained in the order of the letters, the amino acids, the building blocks that make up the protein, okay? And that's a kind of miracle. I mean, I say miracle, but really it's chemistry, okay? And so what do they fold up and what do they look like? Well, actually, they can fold up into completely different shapes. So, for example, this is 3 coils that are kind of wrapped around each other, just like a braid. I grew up in India where women traditionally braid their hair, and that's what it looks like. And so you can have a 3-stranded kind of coil like that, and that's collagen. Some people may know about collagen injections and so on. And collagen is what makes up our skin and connective tissue, our cartilage, and so on. And actually, it's the most abundant protein by weight in a human body. And so it's what gives us our form. Without that, we'd be kind of a blob, a jello-like blob. Now, on the bottom left is a protein, and if you look at these dark objects, they're called hemes. They're special groups that contain an iron atom. And what this protein does is it captures oxygen in our lungs and transports it through our blood to the muscles and tissues where the oxygen is needed. It's called hemoglobin. It's one of the first proteins whose structure was solved. And then this is a protein called rhodopsin. It sits in the membrane of our eye, of the cells in our eyes, and it's what detects light. And when it detects light, it sends a series of signals which then eventually gets transmitted to the brain, and that's how we actually can see something. So what you can see is that here are 3 completely different proteins. They have totally different shapes, and yet every one of them was made somehow by decoding the instructions in our genes. So how does that happen? Well, genes reside on a molecule called DNA. DNA is this long molecule which looks like a double helix, and you can see there are 2 helices wrapped around each other. And just like proteins are built up of amino acids, which are the building blocks of proteins, the building blocks of DNA are called bases, and there are only 4 types of bases. And one of the peculiarities is that each base will chemically pair with only one of the other 4 bases, okay? So a base called C will only pair with a G, and vice versa, or a base called T will only pair with an A, okay? What this means is, if you have one strand with a particular order of bases, it will automatically specify what the order should be in the other strand, because they have to all pair up with each other. And this means that if you have 2 strands that separate, each of them contains the information to make the other strand. And so when this double helical structure of DNA was discovered, and this is Watson and Crick shown next to their model in 1953, they could immediately tell that from one strand, from one copy of DNA, if the 2 strands separate, each could make the other strand, so you'd then have 2 copies. So it showed you how things could replicate, how you could get 2 cells from one cell, so how cells could divide, and how, you could have offspring. So it solved one of the big puzzles of life. But this is the molecule that also contains our genes, and so it somehow contains information on how to make proteins. And that was a big puzzle, how you go from DNA to proteins. And part of the puzzle was solved in the kind of from the 50s and 60s. A lot of molecular biologists figured out how this was solved. And the way that works is that DNA is actually copied into a molecule called RNA. Now, it's not the entire DNA that's copied. You can think of entire DNA as having all of our genes. That would be like a library with all of the books we would want to read. And if you go to the British Library or the Library of Congress, and British Library has every book published in Britain. You go there and say, I want a copy of that, I want to read that book. They're not going to let you take that book out, okay? They're going to give you a copy of the book, either as a digital scan or as a photocopy or a microfilm or something like that. Nowadays, it's a digital scan. So a cell does the same thing. It says, if you want to use a particular gene and read it to make a protein, it makes a copy of that section of DNA, and that's called messenger RNA because it contains the genetic message that needs to be read. And so messenger RNA is like one of the strands of DNA that's been copied. But now that somehow has to be used to make a protein. Somehow the information here has to be used to make a protein. Now remember, this messenger RNA, like DNA, is a chain consisting of 4 types of building blocks. You can think of it as a sentence written in a 4-letter alphabet. Whereas this protein is a chain consisting of 20 types of amino acids. So it's like a 20-letter alphabet. So each protein is a different sentence, if you like. And so how would you go from this sort of 4-letter code to a 20-letter code? Well, it turns out if you just specify that each base on DNA specifies a protein, you'd only get 4 types of amino acids that you could code for. And even if you read them 2 at a time, you'd have 4 times 4, which is 16. It's still not quite enough. So if you read them 3 at a time, then you'd have enough and quite a lot of redundancy built in. And that's what happens. The cell reads the genetic information 3 bases at a time, and that somehow specifies what amino acid to build in a protein. Now this doesn't happen by itself, it happens because there's a small adapter molecule which Francis Crick predicted. It's called a transfer RNA molecule. Because it transfers an amino acid to the growing protein chain. And what it does is it brings amino acid at one end, and at the other end it has 3 bases. And those 3 bases recognize 3 bases on the genetic message, just like the bases across the strands of the DNA pair with each other. So think of it as a pairing scheme. Somehow these 3 bases have to pair correctly with the 3 bases on the genetic message. And if they pair correctly, somehow the system knows to accept this particular tRNA with its amino acid. And similarly, if you get to the next 3 bases, there'll be a completely different tRNA whose 3 bases will pair with these 3 bases. This one, the first one, wouldn't pair there because the bases would be wrong. And so somehow this is the way in which amino acids are brought, recognize, the gene, and then are added to a growing protein chain. Now, of course, this is very complicated, and it's not just going to happen magically by itself. And while molecular biologists were figuring this out, cell biologists, and George Palade, I noticed there's a Palade building here because he spent, the last part of his career here in La Jolla. So George Palade, his lab showed that newly made proteins actually end up in these little blobs. They're not just all over the cell, but they're actually concentrated in these little blobs in a particle, in an organelle inside the cell called the endoplasmic reticulum. And when people then purified these particles, they all looked very, very similar. They were all about 300 angstroms in diameter. That an angstrom is like 1/10 billionth of a meter. Okay, so it's like 0.1 nanometers, and nanometer is a billionth of a meter. So they're about 300 angstroms in diameter, and they all consisted of 2 parts. So one was called the large subunit, and the other was called the small subunit. And somehow these particles, which consisted of these 2 subunits, were reading the gene and making proteins. And that was, the big puzzle. So people then asked, well, what are these particles made of? Well, it turned out the particles were they were very tiny in human terms. You can have 20,000 of these particles across the width of a human hair. So they're really tiny. You couldn't even see them with a microscope. You could only see them with an electron microscope. But in molecular terms, they were enormous. They had 500,000 atoms. and they had 50 different proteins. These are ones in bacteria. The human particles had even are almost twice as large. And they had 50 different proteins, and they were 2/3 RNA by weight. And, this is getting very confusing because you have one kind of RNA that is a copy of DNA, of a section of DNA that brings the genetic message. You have these little small RNAs that bring in the amino acid called transfer RNA. Now you have another RNA which is part of the ribosome itself, which is, making making proteins. And the reason you have all these different types of RNA, we believe, is that life began with RNA. Before there was DNA, before there were proteins, there was an RNA world which could kind of self-replicate and carry on. And then somehow from that world emerged our modern world, which has proteins that do a lot of the action. And the ribosome you can think of as a kind of Trojan horse. It came out of the RNA world, but because it made proteins, it kind of transformed life into the world we know today. So it's this amazingly ancient and important particle. So how would it work? Well, a lot of biochemists figured out that the ribosome binds this genetic message, which, is a copy of DNA, and it has these slots in it. And what these slots are slots to bind those adapter molecules that read the code and bring the amino acid. So it starts off with the first tRNA recognizing the starting place, the first group of 3 bases. And the cell has some clever ways to figure out exactly where to start. So it's brought the first amino acid, and now it's waiting for the second amino acid. And what happens is there's a second slot where another tRNA can come in and bring along its amino acid. Now, if it's the wrong tRNA, it's going to be rejected. That's to say, if the 3 bases on the tRNA don't actually match up with the 3 bases on the gene, then the tRNA is going to be rejected. It won't be accepted. But if it's the right tRNA, the ribosome accepts it. And then when it accepts it, it forms a chemical bond between the first and second amino acid. And this tRNA now has nothing attached to it, no amino acid attached to it. So it's eventually kicked out of the ribosome. And the ribosome then moves over to expose the next 3 bases. And then it selects the correct tRNA for those 3 bases, which brings along the right amino acid. And so then it joins the second and third amino acid. So what it's doing is it's reading the groups of 3 bases along the gene and only choosing the right tRNAs that bring in the right amino acids. And so it's stitching up the amino acids in the order specified by the gene. Okay? So that's the beauty of it. It's translating the language of genes to make the protein that's specified by the gene. So I'm going to switch gears here because this is the state of this was the state of the field when I started studying the problem. And how did I even get to start working on ribosomes? Well, you've heard that when I was 19, I graduated in physics from my local hometown university in Western India, and I took off to go to Ohio University in southeastern Ohio. Part of the reason was that my dad was on sabbatical in Urbana, Illinois, and so normally Indians would get a master's in India and then take the GREs and then apply to schools. I was in a hurry. I thought, well, my parents are there. I might as well spend the summer there, and maybe I should just go to graduate school while I'm at it. But I had not taken the GRE or anything, so I was lucky to get a fellowship anywhere. Most schools wouldn't even look at me. And so I went to Ohio University, which gave me a fellowship, and started doing my PhD there. And I have to say, I spent 5 years there. I had a good time in lots of ways, but my only notable accomplishment in those 5 years was that I met and married my wife. Okay? And and what was the problem? Well, I didn't have any trouble taking my coursework and doing all, getting through my exams or anything. When I started doing research, here's the first problem. I thought, problems in physics were very hard. I don't mean exam problems, okay? I mean, it was very hard to actually make really important advances in physics. And, when I looked at it, there'd been very few really big breakthroughs in the previous few decades. There's a very famous physicist in San Diego named Bernd Matthias who worked on superconductivity. Well, when I was in graduate school, high-temperature superconductors were discovered. People still have no theory to explain high-temperature superconductors, okay? That was 40 years ago, okay? So then, I thought if I stayed on in physics, I would just end up doing boring calculations that would not advance our understanding at all. It might get me tenure, it might get me a job, but, that's not what I went into science to do, to do that sort of stuff. So at the time, I was also reading articles in biology, and I was reading Scientific American, and almost every issue of Scientific American had major breakthroughs in biology. It was, and there was a time when, DNA was being sequenced, structures of molecules were coming out. We were starting to understand what the membranes of cells looked like and how cells were organized. It's just it seemed like a revolution was going on. And I have to say, the revolution hasn't stopped yet. It's still going on. And I knew that many physicists had made this transition before. Max Delbruck, who ended up in Caltech, and Max Perutz, who founded the lab where I work. Francis Crick, who worked, a lot of his career in the lab where I work now, and then came to La Jolla. Actually, the same year I came here, he went to the Salk Institute, and I thought of doing a postdoc. I even had many universities wouldn't accept me as a graduate student, but one or two of them offered me a postdoc. But I thought I didn't know any biology, and if I went to do a postdoc, I would learn a little narrow slice of biology, and I wouldn't end up being a good biologist either. So I thought, I really should go to graduate school again and try and learn some biology. And as I said, most places wouldn't accept me, but luckily, Dan Lindsley, who was the head of chair of biology at UCSD, gave me a break and accepted me with a fellowship. And so I moved, my son had just been born, and when he was 6 weeks old, I and a friend of mine, packed up our belongings and drove from Ohio to San Diego in a Ryder truck. I still remember that ride. So anyway, so here I was in San Diego, and, graduate students are given this sort of, crash course in their first year, and I attended a couple of lectures, and I couldn't understand a damn thing. It was all full of jargon that I didn't know what the hell they were talking about. And so I decided that I actually had to take undergraduate courses. And I talked to Dan Lindsley, and he said, fine. I actually took Dan Lindsley's genetics course, and then I took Paul Price's biochemistry course, and I had cell biology, taught by Rick Fertel and Bill Loomis. And so that was a big grounding in biology that I had in my first year, along with a bunch of rotations. And in the second year, I spent in Mauricio Montal's lab learning how to do actual research. And during the second year, I came across an article by Don Engelman and Peter Moore on using a physical technique to understand the structure of the ribosome. Of course, when I came here, I had no idea what a ribosome was, and that's something I learned while I was here. But then I knew that the ribosome was really important and complex. And I thought, well, if the goal was to come and learn biology, I'd already done that, and I thought maybe I should now I didn't really feel like I needed a second PhD, so I thought maybe I should go off and do a postdoc. And so, I packed up, my wife has been very patient with me for the last 45 years or so, but because every time I say, well, time for us to move, honey, and, she says, okay, fine, and so we packed up and, moved off to New Haven, Connecticut, where I ended up working in Peter Moore's lab. Now, Peter Moore was Jim Watson's student, which had pioneered a lot of work on ribosomes. And they were using neutron scattering to look at where all of the proteins were in the small subunit of the ribosome. Now remember, the ribosome has 50 proteins, and about 20 of them were in the small subunit, and 35 of them or so were in the large subunit. And so the point is, at the time it looked like a great thing to do because the whole ribosome was so huge, nobody had any idea what it looked like. And even just knowing where the proteins were seemed like a good idea. But in hindsight, it actually didn't tell us a whole lot about how the ribosome actually worked. And the reason is, if you want to know how something works, it's not sufficient to have sort of this very low level of information. You have to know what it looks like in detail. And this has been true ever since the history of science. When Galileo saw the moons of Jupiter with the telescope, that changed our view, of the solar system and astronomy. And when Robert Hooke and Van Leeuwenhoek saw microbes and cells, it transformed our view of life, okay? So every time you see a new level of detail, you have a new level of understanding. And that's been true throughout the history of science, and it's been certainly true in terms of understanding how molecules work. So it's a bit like, if you want to know how a car works, if you didn't know what a car looked like and you just saw these dots moving around in straight lines, you're a Martian hovering over the Earth, you'd have some idea that, there's this machine and maybe it consumes gasoline and emits carbon dioxide and water, but you wouldn't really understand how a car works. If you then got a closer look at a car, you'd say, it's got these wheels and that's what it's going around on wheels, but you still wouldn't know how it worked. Then if you opened it up and you saw there was an engine and a steering wheel, and somehow they were all connected, you'd start getting a feel. But it's only when you understand the whole thing in detail. You know the engine has pistons that provide force, and force is generated by combustion of a fuel mixture, and then that force is transmitted through a crankshaft to the wheels, and then wheels turn, and then you have things that control it, and so on. Then you would start understanding, this is how a car works. But to do that, you have to see what a car looks like in detail, okay? And so the question is, how would you see something? Well, if you want to see something very small, you would take a magnifying glass or lens, and what a lens does is collect scattered rays from the object and recombine it to make a much bigger image, and then you can look at the magnified image. This is how we look at small things. And if you are looking at very small things, you use a microscope, which is just a collection of lenses. But there's a very old theorem in physics, which is you can't see detail that's smaller than the wavelength of light that you're using. It's smaller than half the wavelength of light you're using. So if the two objects are closer together than half of the wavelength of the light you're using, you can't see them as two separate objects. They'd just be fuzzy and look like one object. And if you look at the distance between atoms, it's already known that the distance between atoms in a molecule is about 0.2 billionths of a meter, so 0.2 nanometers. And if you look at the wavelength of light, it's about 500 nanometers. So light is about several thousand times larger in wavelength than the distance between atoms. So you can't use light. No matter how powerful a microscope you have, you're not going to be able to look at the structure of molecules. So the way that people got around this was to take molecules, and I won't go through all the details because it's in the book. What you do is you coax molecules to form crystals. And these crystals are basically like regular three-dimensional stacks of molecules. Think of, oranges stacked on in a market, to form a very regular array. And then you take these crystals and you hit them with X-rays, with a beam of X-rays. Why X-rays? Because X-rays have a wavelength of 0.1 nanometers, which is perfect for looking at a molecular structure, because they're comparable to the distance between atoms. And then you would take the scattered X-rays, and somehow you would have to then get information from them. Now remember, the scattered rays from an object that a lens recombines contain the information. The lens is not creating the information That information exists whether the lens is there or not. The lens is simply recombining them to form an image. So these scattered X-rays from the X-rays hitting the crystal must somehow contain information about the structure of the molecule. And so if we could do, and this is what it looks like, so that when you have a crystal, the rays don't scatter in all directions. Like when you look at the screen, it's scattered all through the hall. But when you have crystals, they're only scattered along certain directions because the molecules tend to add up rays along certain directions, and in other directions they don't add up. And so you get these sort of spots. But when you measure that, somehow you ought to be able to combine them. And what you can do is in a computer, you can do what a lens does, which is to say you can simulate this recombining of the rays. And in a computer, you can do the calculation and generate a computer-generated image of the molecule. And that's how the method works. And it's not completely as simple as that, but I've discussed that in some more detail. Now the point is, I didn't know how to use this method, okay? I had no idea how to go about it. And I was at Brookhaven National Lab. When I did my postdoc, I applied for 50 job applications at academic jobs, and I got exactly 0 interviews. And I spent a disastrous year in Oak Ridge National Lab under a misunderstanding. And then I ended up leaving and went off to Brookhaven National Lab, where I did have the opportunity to do research. But I didn't know any crystallography, and I was stuck, with this technique of neutron scattering, which really wasn't going anywhere. It's a great technique for certain areas of physics, but in biology it's not been very useful. So when the time came to give me tenure, they said, well, what will you do if we give you tenure? So usually they expect you to say, oh, I'm going to do this, 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 and this, along the lines of my work. And I said, well, if you give me tenure, the first thing I'm going to do is stop doing what I'm doing and go on sabbatical, to learn crystallography. And luckily, they were far-sighted enough that they said, fine, go ahead and do it. And so I wrote to, so then the question was where to go. And, my wife and I used to love watching masterpiece theater and so on. So we thought it'd be great to go spend a year in England. And also, it was the birthplace of this whole technique of crystallography. Several Nobel Prizes had been won for using this technique. And the director at the time was Aaron Klug, who was a very famous guy. He was a Nobel laureate and went on to be, ironically, he went on to be president of the Royal Society himself. And I was a sort of no-name, scientist working at Brookhaven National Lab. So I wrote to him and I said, look, I want to come there and learn crystallography. And by the way, I've crystallized this protein that you might be interested in. So that was the hook. I'd crystallized something that he cared about. And so instead of throwing my letter in the wastepaper basket, he offered to sponsor me for a Guggenheim Fellowship. And I was off to the races and went there. And it completely changed my view of the world and changed my view of how to do science, okay? And I'll talk about that later in the talk. So I came back from my sabbatical, and I thought the whole ribosome seemed really too hard, although I had some ideas about how to do it, and as a result of my sabbatical. And so then my colleagues at Brookhaven, Steve White, who then went off to Duke University, and now he's at St. Jude in Memphis. So we started to look at pieces of the ribosome, and so each one, we take each protein, try and get crystals of it, then solve its structure and figure out what it looked like. Now, this seemed like a good thing for a while, and certainly it was good enough that NIH was convinced to give us grants for, at least a decade and a half. But in the end, it turned out it started looking like a stamp collecting exercise because, it'd be like if you want to understand how a car works. Instead of figuring out how a car works, it'd be like you went to an automobile parts store and you say, I want to look and see what a spark plug looks like. I want to see what a fuel injector looks like or a hubcap looks like. Okay, you know what these things look like, but you have no idea where they even fit in a car. You have no idea what a car looks like, so you don't know where these parts go. Okay? So you're not getting any closer to understanding how this big machine, the ribosome, works. So things started to change when this guy, Heinz Gunther Wittmann, who was a Max Planck director in Berlin. Now, he was always interested in, trying to get a structure of the ribosome. And he had a couple of false starts. There are two very funny stories in the book about, the first two attempts that people that he tried to bring in. One turned out to be a complete fraud. Another wanted to go to Germany because his girlfriend was German, and then she dumped him, so he didn't go. Okay, but then the third person to write to him was this Israeli scientist, Ada Yonat, and she said she wanted to come and spend a year in his lab. And so the two of them started to collaborate, and they got crystals of the large subunit of the ribosome. And then a few years later, a group in Russia obtained crystals of the small subunit of the ribosome and also of the entire ribosome. So now groups had crystallized both the small and the large subunit and the whole ribosome. So, you'd think that, okay, now things are going to happen. But that was around the first crystals were in 1980, and then the Russian group's crystals were around 1986 and 87. But by the mid-1990s, we had a pretty good idea of what the ribosome did and a rough idea of its shape. But 15 years later, after the first crystals, there was still no progress towards an actual structure of the ribosome. Okay? It looked like the field had sort of stalled. And there were, by that time, the crystals vary in quality. Not all of them can give you an atomic structure. For that, the molecules in the crystal have to be very precisely aligned. But after several years of work, Ada Yonat's group had produced 50S crystals that really had good enough quality, and I explain in the book what that means. But many years after that, there was still no structure, not even a sort of low-resolution structure. So after a couple of meetings, when it looked like, her group was stalled, the Yale Group, headed by Tom Stites, along with my former mentor, Peter Moore, decided that, well, they can't just wait around and they're going to give it a crack. And so they took Ada Yonat's crystals and decided to see if they could somehow make progress with those crystals. Now, I didn't want to do that. I thought, well, I didn't know how the community would react if I did that. And I also thought, there are already two groups now working on these large subunit crystals, so let them, fight it out. And I didn't want to get involved in this three-way competition. But there weren't any good crystals of the small subunit, so I thought maybe, and there weren't any good crystals of the large subunit, so I thought maybe I can take the small subunit and try and get crystals that were good enough to get an atomic resolution structure. And I thought the whole ribosome was really way too hard and would have to wait until we'd figured out how to solve these subunit structures. So I started to do that, and I was a professor at the University of Utah then, and I had, and you can see this is a picture taken, this is in my Bin Laden phase, okay? And you can see that I'm the only guy who was wearing lab gloves at the time. So you can see that when this picture was taken, I was the only guy doing any work, okay? But anyway, and in those days you could actually, before health and safety guys got really agitated, I could even bring my bicycle and put it just right next to my lab bench. Okay, so these were two graduate students. This is Bill Clemens, who's now a tenured professor at Caltech, and this is John McCutcheon, who's also a tenured professor in Montana. And so these two guys knew nothing about either ribosomes or crystallography, okay? And I said to them, it's a good idea, crystallizing and solving the structure of this 30S subunit, which is, several 100,000 atoms, okay, is a great idea for a PhD project, okay? It was about 5 times larger than anything that had been solved. And so they said, great, let's do it, And it shows you that if you know too much, this guy, Bryan Wimberly, was a very talented postdoc who turned out to be incredibly important for the structure. But he was a little reluctant. It was his second postdoc. He was reluctant to join this effort right at the beginning. And it shows you if you know too much, it's sometimes a dangerous thing because you know all the ways that it won't work and all the difficulties and so on. And if you don't know enough, you just say, oh, great, I'm going to do it, and just start going, going for it. Okay? So they started off, we started making progress. And then I got a huge shock. I was invited to a meeting in Sweden, and there I found the Yale Group was making progress on the large subunit. And then I found to my shock that Ada Yonat had given up on the large subunit. She basically said it was unworkable. But in the meantime, she had found a condition in which her small subunit crystals, which were not diffracting, not of good enough quality, were now sufficiently good quality. So it's clear she had shifted her focus from the large subunit to the small subunit. So suddenly, instead of Yale Group and Ada Yonat duking it out, I found that I was going to be in a head-to-head race with her, okay? And she had this very established group. She'd been doing it for 15 years. She had 2 labs in Germany and a lab at the Weizmann Institute in Israel. And I had my 2 first-year graduate students in Utah, okay? And so on the way back from this, on the plane ride back from Sweden to Utah, I thought, what the hell am I doing? Should I, maybe I should give up. And then I thought, well, actually, this is the most important thing in my field, and if I don't do it, I'll regret it. But if I give it a shot and I fail, well, then, at least you've given it a good shot. And I said, I also thought, crystals have been around for 15 years and nobody's solved it yet, so, why should I think that they're going to solve it tomorrow? If I just keep plugging away, and I had some good ideas of how to do it, which I describe in the book, then maybe, it's going to happen. So then, I found out that there's a famous ribosome scientist in my field called Harry Noller, who was really a biochemist. Paul Sigler, who was a famous crystallographer, he didn't like biologists referring to him as a crystallographer because he said crystallography is just a technique. We're actually biochemists trying to understand problems. We're not, defined by technique. And so in revenge, he would call these biochemists gel biologists because they'd all run gels, this electrophoresis gels, So Harry Noller was what we used to refer as a gel biologist, okay? He ran hundreds of gels, okay? And so suddenly I found when he visited Utah that he was working on the structure of the entire ribosome. And I thought, how is that possible? He's not a crystallographer. And it turned out that he had imported these 2 people from the original Russian group that had crystallized the entire ribosome. And he'd also hired as a postdoc this hotshot young crystallographer, J.B. Cade, who's a brilliant guy who's now at Berkeley. And so he had assembled a crack team to try and work on the whole ribosome. So suddenly, my quiet niche on the small subunit had become like a 4-way race, okay? And so it was really stressful. And then I did something that most people thought was completely crazy. If you're in the middle of a race, the last thing you want to do is move. Because everybody knows that if you move, it's going to cost you at least a year, okay? But I thought, I was looking sort of long-term, and so I thought, why should I move? So I wanted to work on the whole ribosome structure, and I didn't know how long it would take, because 15 years had gone by since the first crystals. It could be tomorrow, it could be, another 5-10 years, I had no idea. And so I thought I needed to go in a place which had stable funding. Now, at Utah, like at UCSD or anywhere else, you're funded on research grants. You get these 3, 4, 5-year grants. Now, if you have a 5-year grant, and at the end of year 4, when you have to submit your renewal application, if you haven't made your breakthrough, you're in deep trouble, okay? And especially if you haven't done anything else. And I couldn't afford to do anything else. This was a problem that I was going to have to completely focus on, especially with the competition and because it was so hard. So I wanted to go a place where they wouldn't pull the rug out from under you, just simply because you hadn't made your breakthrough, as long as you were not spinning your wheels and were actually making progress. And so I also thought, I want to go to a place where people really understand how to push the limits of the method. So what we were doing was beyond the limits of what had been done using the crystallographic technique. And it's a little bit like, if you want to drive home in your car, no problem. You don't have to be an automobile engineer or a car mechanic. But let's say you want to drive across the desert in your car, okay, the Sahara Desert. You better either know everything about how a car works when it breaks down, or you better take along a couple of really good mechanics with you, okay? So I thought, this is more like driving across the Sahara, so I better be in a place where if things run into problems, I have really expert colleagues. As it turned out, I didn't end up needing them, but if I had needed them, they were around in case I wanted to talk to them. Another is, this is a place where it has a very collegial atmosphere. People work in very small groups. Young scientists are given complete freedom to do what they want. They have the same rights and the same resources as established groups. Whereas, in most places, a young group has far fewer resources than a very established group. And there's no hierarchy. Everybody sits in the same tables in the cafeteria. The electricians and the cleaners and plumbers sit with the lab director and the first-year students. And it creates a kind of atmosphere that we're all in this together. And finally, it's an institution with a long tradition of tackling very hard problems with very small groups. The groups are typically 5 or 6 people. 8 is a very large number, okay? And the emphasis is always on how important is your problem. Is your problem, if you solve it, is it going to advance the field? Not, is it going to result in your next paper, okay? And somebody like Fred Sanger, who got 2 Nobel Prizes, by today's standards, if you look at it, he had very few papers, in his life. But 2 Nobel Prizes transformed molecular biology. And so not surprisingly, the lab has got its 15th and 16th Nobel laureate in the last 2 years. And if you look at them and ask, how long did it take? Well, they started their work 25 or 30 years ago, okay? And they were, initially, little progress, and they were just funded, and as long as they were making progress, they were supported. So my wife had been moving around. You saw they moved from Ohio to San Diego to New Haven to Tennessee to Long Island to Utah. And so finally I said, look, we really have to move again. And by the way, I'm going to want to take a 40% pay cut to go live in a country that's about 20% or 30% more expensive. Okay? And so she said, okay, I'll do it, but this is the last time I'm going to move. And any time I get an inquiry from somebody, it doesn't last more than a day because my wife says, no, we're not moving again. And so we packed up our belongings and moved to Cambridge. And I describe in the book, we had made our initial breakthrough in Utah, but to get to the next stage proved incredibly hard because of all kinds of technical problems that I describe in the book. And also I had to partially assemble a new group because only 2 people moved with me from Utah, and I had to hire these 2 guys who joined my lab without even seeing me, okay, just by emails, and it was a tough problem. So when people say, you showed great courage, that's nonsense. I had tenure, okay? If it had failed, I'd still have a job. These are the guys who were risking their careers by, coming to my lab and working on it. And, this is Bill Clemens again, who came with me to Utah. And then we finally were able to crack the structure. And this is Bryan Wimberly, who spent his entire year and a half, he had moved with me from Utah, he spent his entire year and a half in this dark room trying to figure out what the structure looked like and, helping the rest of us, guiding the rest of us in terms of building the structure. And then we also had to use very powerful sources of X-rays. So here we were in Cambridge, and we had to, go off to southeastern France or to Long Island or to Chicago or to a synchrotron in the north of England. Wherever we could get beam time and X-ray time, we would have to ship all our crystals. And there's a lot of funny stories about that. And this is the beamline that helped us crack the problem. It's the beamline in Chicago at something called the APS, or the Advanced Photon Source. So ultimately, the whole thing worked. And after 48 hours of data collection, so we all worked in shifts around the clock. We did a calculation, and it looked like the experiment hadn't worked, and I was really depressed. And then I realized we'd made a mistake in the script that we had submitted to the computer, and when we corrected the mistake, it spat out these enormous peaks that showed, that the entire experiment had worked. And after 48 hours without sleep and several months, almost a year of stress, suddenly it was such a relief that I jumped out of my seat and started dancing around the office, saying, we're going to be famous. And my technician thought I was completely crazy. He didn't know what to think. So when the structure works, you don't get the structure. You don't know where the atoms are. And this is like solving a complicated jigsaw puzzle. And what it is you get a three-dimensional image of the molecule, and you have to somehow figure out what the structure is. It's a little bit like solving a three-dimensional jigsaw puzzle. And just like with a jigsaw puzzle, what you would do is you would look around and look for patterns you recognize. So you'd poke around the structure, and then you'd see, oh, look at that. Here are two strands that seem to be locked into each other. And if you know what you're doing, you'd recognize that as a piece of double helical RNA. So RNA can also form double helices just like DNA. And so you suddenly have taken a piece of that structure and interpreted it in molecular terms. And so suddenly you've got a little piece of the structure in terms of atoms, from the image by sort of interpreting it. And so then you start looking for other pieces of the structure that you can recognize and then start fitting in, atomic structures into them. And so you keep on doing this until you've solved the entire structure. And so then when you've done that, you sort of declare the structure solved, and then you go off and publish it. And so doing that, while we were doing the small subunit structure, the Yale Group, in fact, they were slightly ahead of us, solved the structure of the large subunit. And these two structures both came out in the summer of 2000. And they told us a lot about how the ribosome worked, including showing evidence that the ribosome probably came from an ancient RNA world because they're fundamentally RNA machines. And then it took us another 6 years before we could solve the structure of the entire ribosome with, those tRNA molecules that bring in the amino acids together. And this structure has about 500,000 atoms, and at the time it was the largest structure solved using this technique. And one fringe benefit is that the ribosome is one of the, as I said, it comes from this ancient form of life. So it's so old that during evolution it's diverged a lot. So our ribosomes are, even though the core looks very similar, it's diverged from the ribosomes of bacteria. And so the result is that there are compounds that can bind bacterial ribosomes without binding to our ribosomes. So they can stop bacteria from working because the bacteria won't be able to make proteins. And proteins, as you know, do lots of things in life, most of the things in life. So if you stop something from making proteins, you basically kill it. So these are useful antibiotics, the ones that bind only to bacterial ribosomes but don't bind to our ribosomes. And many of them were discovered in the 40s and 50s, but people still didn't know exactly how they bound to ribosomes because there's no structure of a ribosome. But once there were structures of ribosomes, we could easily figure out how the antibiotics bound to the ribosomes. And this is one such antibiotic. This is spectrinomycin, and people actually have used these structures to design better analogs of spectrinomycin that are effective against Mycobacterium tuberculosis. Of course, going from there to useful medicine is a long path, but it shows you how you can use these structures to try and design better antibiotics. And so we went on to do a lot of different antibiotic structures, and other groups did many more antibiotic structures. So another part of the book is, so that's about the ribosome story. Another part of the book is the human side of science. So scientists are neither Spocks, we're not like these emotionless, completely irrational beings, half Vulcans, if you like, and nor are we saints, in the business for altruism and the, noble quest for the truth and helping humanity. It's, it's somewhere we're complex. We're motivated by curiosity. We're motivated by ambition, by desire to succeed, to be first. We have egos, we have insecurity, we have anxiety. We have rivalry and competition. Sometimes we're competing and collaborating at the same time, depending on what's best for us. And sometimes even with the same person, we're collaborating on one project while competing with them on a different project, okay? And so it makes life very interesting. And, but it's not all about competition. People also helped me in many ways. For example, many people gave me advice all throughout my life, and especially in this process. And for the ribosome, many people gave me reagents that are not commercially available, that were really very specialized reagents that they could only make because they were experts. And none of them, not a single one of them, wanted to be a co-author on any of our papers. They just gave us reagents just to help science along, okay? And, many people also gave me technical help. And so all of this with no expectation of anything. So you see the full spectrum of humanity. You see some of the ugly side of humanity, all this jealousy and rivalry and backbiting and so on. And you also see the great side, people helping you with no expectation in return. As, science is just a microcosm of life. And so the book goes into, what it's actually like to be a scientist and to do science. And then, you start throwing prizes into the mix. So, this corruption starts early in life. Even in school, you're starting getting prizes, being first in your class or whatever. And then, being valedictorian, you get a little medal. And then, you start getting early career awards, you get some prizes all through graduate school. Then you start your job. You start getting early career awards. We all know all these fast-track scientists who all get all these racking up awards as they go along and from graduate school, early career. I have to say, I was completely award-free until I was in my 50s. So and then you start getting. Then they start getting awards. Then they want to get elected to their national academies. They get annoyed if they're not elected to the national academies. Once they're elected to the national academies, they're still not happy. They want to get bigger prizes, And the whole thing is corrupting, and it's because it's applying a sports metaphor to science. So if you have a 100-meter race, first of all, the rules are very defined. It's a fixed distance. You've got to start when the gun goes off and not before. And, at the end, there's a camera that figures out who came first, second, and third, okay? But science isn't like that. Science is multidimensional. It's not clear which idea was the most important, sometimes very subjective. In a field called transcription, one big prize, the Lasker Award, went to one guy, and the Nobel Prize went to a completely different guy. How is that possible, And so, it can be very subjective. And the other thing is the Nobel is given to 3 people, and no more than 3 people. Often, more than 3 people contribute important ideas or do important work in a field. It's not like a gold, silver, and bronze, So what this does is it sort of warps people when they're in that, so I call these diseases pre- and post-Nobelitis. So when people have done very important work, a lot of people will tell them, this work could get a Nobel Prize someday. And so suddenly they start thinking, well, maybe it could, And then suddenly, then they're starting to do this rule of 3. Well, if I'm in the running, who are the other people in the running, And are there more than 3? And, then they start talking up their work and maybe talking down other people's work, So it becomes like a political campaign. And, so I talk in the book about how people behave. And so after these structures came out, there's a chapter called the Ribosome Roadshow, because all these people, who are sort of in the, running, they should be enjoying life, okay? But instead they're worried about competition, and who else is in the running. And so they're all invited to conferences all over the world, and it's almost like a political campaign. They're jockeying for the 3 chairs left when the music stops, okay? And, suddenly a bunch of us start getting invited to Sweden. Now, of course, we get invited to meetings in Sweden. There are a bunch of people invited, and some of them are people the Swedes are looking at, to consider for a prize, and other people are decoys, Well, there are people in the field that they've invited to sort of, have a nice conference. And so when you go there, you're never sure whether you're one of the contenders or whether you're a decoy, okay? And so you're trying to do your best and give your talk. And at the last meeting I went, it was on the central dogma, which is how DNA replicates, how it's converted to RNA, and how RNA gets converted to protein. And so a bunch of us were in this field, in these fields, and, there were some great scientists there. And all of them, many of them were really nervous. One of them was hyperventilating, So it's like these grad students before a prelim exam, And here they are, like you can see they're sort of auditioning. And so I go into the book very, very frankly. Of course, I don't mention names except when it's okay. But I don't mention the name of the guy who was hyperventilating. Anyway, and they're trying to be nice to people on the committee and so on. And then at the end of the meeting, there was a banquet. The last evening of the meeting, there's always a banquet. Scientists like to sort of chill out and after all the stress. So at this banquet, a Swedish ribosome biochemist, who was the only Swedish speaker at the meeting, came up to me, and he disagreed with one aspect of our work on the ribosome, which is how the code is read accurately. And he started getting into a big fight with me. And, initially I was sort of polite, but then I started fighting back. And then, his colleague had to come and separate us, And then 2 months later, I found out that the guy had been appointed to the Nobel Committee for Chemistry. So after that, from 2004 to 2009, I declined all invitations to Sweden because I thought, why the hell should I go and eat bad vegetarian food in Sweden when I'm not even in the running, because this guy, as long as this guy's on the committee, there's no way that I'm going to get the prize, okay? So why should I even bother? So then it turned out, one October in 2009, I got this phone call, and I was so convinced I was not going to get the prize. I was sure it was a prank. I have a couple of friends who I thought would play that kind of prank. So I told the guy, you have a really great Swedish accent, but I don't believe you. And then, so then it turned out that the guy. So then I said, okay, if it's true, then I know Måns Ehrenberg's on the committee, so I want to talk to him. And Måns Ehrenberg came on. He was the guy who had a fight with me, and he's he congratulated me and said, you really deserve it, but it's the last time I'm going to tell you that because I still don't agree with you about it. But it showed you that the guy had terrific integrity. He disagreed with me about something that we were both working on, but he put that aside and looked at the bigger picture. And, because it would have been very easy for him to say, Ramakrishnan, he's okay, but he's not that great. And that would have been enough to sink me, And so it really tells you something about the Swedes. One of the reasons the Nobel's respected is because it, I think whether you agree with their decision or not, they're really honest and they have integrity. So if you look at my weird career and all the twists and turns and all the changes I've had to make, I never thought I would end up in this place. And this is a banquet after the prize. And just to give you an idea, this is the King of Sweden. So all the dignitaries and the laureates are seated on this long central table. So this is the King of Sweden, and next to him is Ada Jonat, with whom I shared the prize. And next to her is me, and then this is the Crown Princess Victoria, and on the other side is Tom Steitz, with whom I shared the prize. And my wife Vera is over here in animated discussion with the German Vice Chancellor. And so, there are 3 Science Nobel Prizes, and there are, kind of what I call a pseudo Nobel Prize in economics, and then there is honestly, it was it's called the Swedish Riksbank Prize in honor of Alfred Nobel. It was only instituted in the 70s. It was not Alfred Nobel's original will, and then there's a literature prize. But of all things, the press really understood that the really important thing that year was the ribosome, because the next day in the morning, they put Tom's tights and me on the front page of their newspapers, okay? And so they clearly knew that it's the ribosome that's important. And the fact that Princess Victoria was between us had absolutely nothing to do with their choice of photograph, okay? So I want to close with some general lessons, which is, looking back at my life, I've had a very weird career. I went to, not so prestigious schools. I started off as a physicist, gave up on it, tried to learn biology, for which I came to UCSD. But then I did a postdoc using a technique that turned out to be a dead end, and then I had to go off and learn, a technique that was useful. And then also I had to move different times to keep giving myself opportunities to do the kind of science I wanted, sometimes even taking a huge salary cut in the middle of a big race, okay? So the lesson from that is you've got to keep your options open. If you don't keep your options open, you'll find yourself out of the game. You won't be able to play the game, okay? And then the other lesson I learned on sabbatical is that I found great scientists like Max Perutz or Aaron Klug asking the dumbest questions after talks, okay? And made me realize, actually, the questions aren't dumb. They just didn't know the answer, so they asked it, okay? And so it shows you should never be ashamed to show your ignorance, because the only way you can get out of your ignorance is to ask, and so you have to show your ignorance. And likewise, you shouldn't be afraid to ask for help when you need it. You can't pretend that you know stuff that you don't know. And then you have to talk to people. So the idea for how to solve the ribosome structure came out of a random conversation I had with a colleague who's now a very good friend of mine while I was on sabbatical. And that idea sort of stayed in my brain and for many years, and then eventually, I had the courage to then take the plunge and work on the whole ribosome. But you can't talk to people all the time, because then you'll develop a pack mentality and you won't be able to think originally. You've got to have your own ideas and mull over them and really obsess over them. And then another thing I realized is, everywhere I went, I thought, oh my God, these people around me are so smart, and they're going to find out how dumb I am, and I'm going to be exposed. And most of us hope that, by the time that happens, we'll have retired. And so this is called imposter syndrome. Okay, and the fact is, everybody has it, and even the people you think are smart, they have it too. But sometimes if you're an outsider, and you can be an outsider because you're geographically an outsider, or because you've been to not so great universities, or because you're an ethnic minority, or because you're a woman, because there are not as many women in science. So, you can have, if you're a minority of some kind, you feel this imposter syndrome even more. Because you think, oh, these people are all a little bit different from me, and, they're all smarter than I am, so that's the sort of feeling you get. And so you've got to realize everybody has it unless they're sociopaths, in which case you don't have to worry about them. And so you should realize that and just not let it bother you. And then science doesn't, people think you have to be a genius to be a scientist, or you have a lot of innate talent. But in fact, there are other things which are much more important. First of all, you need money, because science, at least you need to have a salary. Even if you're a theorist or mathematician, you need to have a salary to be able to do your work, or you need money for your research. And then you need to have skill, which requires training and proper education and background. And, if you're working on anything worthwhile, it's going to be hard. Most of the time things are not working, because as soon as things work in science, you move on to the next step. Then again, things are not working. So you've got to have patience if you want to do science. And then you always need that bit of luck. And most of us have had luck. Many people try to forget about the luck that they had, and then in hindsight, it's all their cleverness. But actually, even talking to a random person who gave you an idea, even being in the right place at the right time, having the right mentor, having the right colleagues, there's a lot of luck involved in science. So I'd like to stop there, and thank you very much for your attention.
Okay, well, thank you for that beautiful talk. It's so important for all of us to recognize the troubles, and especially for our younger generation of students to recognize that it's not a straight path to the Nobel Prize. So thank you so much for sharing so much of that. I think that it's...
I forgot to mention one important thing about the Nobel Prize. That is, one of the reasons it has so much cachet is because in the general public's mind, the Nobel has some special, aura to it. And they think if you're a Nobel laureate, you're some kind of genius. And I point out in the book that the Nobel Prize is given for making an important discovery or an important advance. But I also point out it's a bit like Shakespeare said in Twelfth Night, that some are born great, some achieve greatness, and some have greatness thrust upon them. It's the same thing with Nobel laureates. Some of them are very smart, some of them just happen to be in the right place and keep persisting, and some of them simply have stumbled on to something important, and lucked out, okay? And maybe they're not even, actually, I won't name any names, but some of them are not even that smart, okay? And also, outside their own field, Nobel laureates they're really no better than anybody else. And so I call these diseases pre-Nobelitis, where, you're anxious about the Nobel Prize and, whether you'll get it or not. And then post-Nobelitis is, despite their, whatever it is, when the public starts, adoring them and, praising them, it goes to their head. And pretty soon, they start pontificating about all sorts of things, and they spend all their lives, traveling from one airport to another and so on. So that's called post-Nobelitis, and I try to avoid both diseases.
I'll say you've done a beautiful job. Your research is continuing on. I've enjoyed my time with you. So do you have advice for students or young scientists about how to pick the right problem moving forward? Because you want to be at the right place on the curve if you're to achieve greatness.
I think, first of all, you should completely ignore prizes and awards and things. Considering them as a complete recipe for unhappiness, because Nobel Prize is a bit like a lottery. There's so much good science going on, and it's given to one prize a year to 3 people. It's pretty much become a bit of a lottery, okay? But what you should do is pick a problem that you really care about, because most of the time in science, nothing is working, okay? I can tell you that, because as soon as it works, you publish and you move on, or you move on to the next step, and then again nothing is working. Because if you knew how to do it, it wouldn't be research, right? So 90% of the time, it's not working, okay? And so you have to care enough about the answer, about the question, to want to come in every day and give it a new try, try something else, or try some new thing. So you've got to care about the question. So I would say, pick something you really think is really interesting. Don't worry about what the rest of the world thinks is interesting, because I can tell you, when I joined UCSD, cloning was all the rage. There's a very famous scientist here named Don Helinski, and everybody wanted to join his lab, okay? Now, 10 years later, everybody is cloning. Now, you order your clones. You don't even make clones, okay? So, it goes to show you, if something is really fashionable now, by the time you've spent 5 years of grad school and, you've done your postdoc, it'll be 10 years before, you're sort of starting your career, that the problem might have been solved or might not be fashionable anymore. So just pick something you like and then just work on that. And then that way you'll care about it no matter whether styles come and go.
That's great. Thank you.
Yeah.
I think we have a question here on the right.
Well, I'm number one here. My name is Chris Wills. I'm an evolutionary biologist here at UCSD. And let me ask you an evolutionary question, if I may. Can you hear me? Yeah, good. The one neat thing about the ribosome is that all the business parts of the ribosome are, in fact, RNA. But the ribosome needs proteins for it to function, because if you take the proteins away, it falls apart. Somewhere out in the great tree of life, are there ribosomes that need fewer proteins, that, for example, or need proteins that, have other functions and so on? How is that story emerging?
Yeah, so this is a chicken or egg problem, which is if ribosomes are the things that make proteins, and they themselves have proteins, how did the ribosomes even come into being? And Francis Crick, in the late 60s, was the first person to have this insight where he said, maybe early ribosomes consisted only of RNA. And somehow the system got going and they started making proteins, and then as they evolved, they made proteins that stuck to themselves, and that's how we ended up with the modern ribosome. And Harry Noller was one of the people who really pushed this idea biochemically. But biochemically, he could never completely prove that the business part of the ribosome was all done by RNA. That came out only when the structures came out, when the large subunit and the small subunit structures came out. But even today, nobody's created an entire ribosome that's only made up of RNA. And even if they did, doesn't mean that that's what existed before. But there's no other way it could have come into being. It had to be RNA. We still know that the core of the ribosome is all RNA, so probably the earliest ribosomes were just that core with the RNA, and then the proteins came later.
Okay, that's beautiful. So I was wondering if you could say a few words about the role of technological innovations in discovery.
So one thing I point out in the book is, we think of scientists as somehow, these geniuses who just completely made this huge leap. And in fact, it doesn't work that way. What happens is that the state of understanding in a field gets to a point where a few people realize that the next step is possible. Sometimes it's a level of understanding, and sometimes it's a level of technology. So if you look at calculus, okay, you had centuries go by, there's no calculus. Suddenly, two people within the same lifetime, Newton and Leibniz, invent calculus, okay? How could that be? Same thing with evolution, theory of evolution by natural selection. You have Darwin and Wallace, within their lifetime, come up with this theory of evolution. You get many such things. Now, the ribosome, stalled for a long time, suddenly 4 groups are working on it. What happened? Well, synchrotrons were available, fast X-ray detectors were available, this technique for solving the structure which was based on synchrotrons were available. So few people see that and say, aha, now that we have this, we can do this next step. So science is driven by a combination of a level of understanding and the level of technology. And so technology drives science, science then results in new technology, which then drives new science. So it's this highly interconnected web. It's not that you have science which generates technology. It's all circles back. Technology also drives science.
That's beautiful. Thank you. Do we have questions?
Hi, I'm Dwayne Stupak, and I'm in the division of gynecologic oncology. I have a lab over in the Cancer Center. And so we deal with survivors all the time.
Sorry?
We deal with cancer survivors all the time, and we try and explain the science we're doing to them. And one of the things that I really love is the way you're communicating science in a way that the average person can understand. And so I think the book comes at a very timely time because if you look at what's happening right now, we're living in an environment where the public is increasingly dissociated from science. They trust scientists less and less. And I don't think that we're going to be able to shut down the internet overnight. So what are the steps that you think we can begin to do to communicate our science better to the public and really build bridges?
Yeah. So I want to slightly disagree with you about the public and trust in science. Certainly in Britain, where they did surveys, it turns out scientists are among one of the most trusted professions. And by the way, journalists and politicians are among the least trusted professions, okay? And lawyers are somewhere in between. And so But it is a serious problem in an era of fake news and important problems like GM crops or climate change and so on. You can have all sorts of views on the internet. Vaccination, a classic example. There's anti-vaccination movement, okay, and which has already cost thousands of lives actually. Yeah. So. So I think it's very important for scientists to be able to communicate regularly with the public. And it starts with going to schools and explaining to children how science works. And part of the problem is the public wants to know yes or no answer to everything. And we have to communicate that in science there's always uncertainty. And even the truths in science are provisional truths. They're what we believe now, but if tomorrow evidence comes along, then we're willing to change our minds. It's not like frozen. It's not like a dogma or a religious belief. It's based on evidence. And so we have to communicate how science works, that it's based on evidence, that it's provisional, that it can change. And then we have to communicate what is the evidence for all these things and why do we believe what we believe today. And you've got to start with schools, you've got to start by, with public lectures and so on. And many scientists don't want to do that. They're too busy, doing their work, writing their next paper, getting their next grant. But it's really, we're funded by taxpayers for the most part. So it's really part of our duty. And now I wouldn't want every scientist to go to the public, because I can tell you, many of my colleagues, they're better off just working in their lab and not being exposed to the public. But those scientists who are able to communicate and have an interest, we should not penalize them for doing that. We should encourage that. And part of the thing is the reward system in academia only measures, it often doesn't even measure how good they are as teachers, okay? I mean, the sad thing is, it measures how good their papers are and how much grants they pull in, even if they're doing a superb job of teaching undergraduates, that doesn't seem to count as much. And that's not right, okay? So we've got to balance the way we evaluate scientists and give them credit for teaching, credit for public engagement, and credit for research. So the system needs to have more balance.
Just as a follow-up, do you think that there's more outreach in the UK than we're doing here in the US?
Sorry?
As a follow-up, do you think that you're doing more outreach to the community than in the UK than we're doing currently in the US?
I don't know, because when I left, I was pretty much a lab rat the entire time I was in the US, and even most of my life in the UK. But it's only in the last 10 years, and especially in the last 3 years when I became president of the Royal Society, that I took on a much more public view and started to appreciate the importance of all these things. So I'm, if you like, a late learner to this process. So I'm just as guilty of it as many scientists are. So I couldn't tell if there's more in the UK. But in the UK, we do have things like the BBC, which is a fantastic outlet for science programs. In fact, if you watch NOVA or many PBS programs, many of them are really produced by the BBC, So we do have a tradition of David Attenborough, for example. So we do have a tradition of public engagement in Britain.
Great, thank you. One more.
Hi, I'm Terry Gasterland. I'm a professor here at UCSD. I'm over here on here,
Hi.
Right. Back in 1996, I had the pleasure of working with Harry Noller when I was at Argonne National Lab.
Oh, yes.
2 years after coming back from 6 months in Pushno, Russia. So I had all these memories as you gave your talk. With Harry, we put the ribosome in the cave, the immersive computer visualization environment.
Sorry?
And we put the ribosomal structure
Yeah.
in a three-dimensional immersive environment. And with Harry Noller, we walked around in the ribosome and looked at the positions in the ribosome that were fast-changing versus slow-changing,
Yes.
and which ones were on the surface versus not on the surface. Well, for Harry, he went back to the lab in Santa Cruz and started doing all sorts of things. But what was inspiring for me was walking around in the ribosome launched me on my career in computational genomics.
Yeah.
And I'm wondering whether there have been moments when visualization of data has launched you on a new step.
Oh, I think visualization of data is incredibly important. And, one of the problems is with these very complex structures, trying to get, humans are not used to looking at, we didn't evolve to look at screens. We really evolved to, have a three-dimensional view of the world, to be able to touch and tactile and so on. So I think, and the worst part is we're still reading papers which are essentially two-dimensional still representations, very, very complex data. So I think visualization of data is really a big blossoming field. And with new techniques, 3D printing, virtual reality, and so on, we're going to be able to try and understand things much, much better than we did, in the past.
Thanks.
That's great. Thank you. Okay, do we have one more question over here?
Hello, my name is Irina. I'm an undergraduate student here, and I read a lot of nonfiction, not so many autobiographies, but I really loved how you combined your interest for ribosomes and the science of politics, and especially how much you've attributed, like how much you appreciate your collaborators, your graduate students. I thought it was pretty unique for writing of autobiographies, at least in my experience. And I was wondering what inspired you to combine all of these issues in one book, and what other books I could read that also do that.
So I wanted to tell a very frank story, okay? Now, most science books are very, very sanitized, so they have this sort of very linear path, and in hindsight, everything is great and so on. Now, one book I read when I was, I was about 17, when it came to India, was The Double Helix by Jim Watson. We all know what Jim Watson's reputation is today, but when that book came out, it was very different from any other book, because it told you what it was like, to be in the middle of the story, to be in the middle of the action. It was the first book that tried to be completely brutally honest about what it was like. Perhaps too much so, because he was unnecessarily, obnoxious in the book about, personal appearances and, things like that, which were not really necessary. It wouldn't have detracted if he had left out some of those parts. So what I wanted to do was capture that honesty of that book, but not go over the line like he had. And luckily for me, Richard Dawkins, who did a review of my book, described me as a sort of nice Jim Watson. So I'm very glad he added the word nice, okay? Because otherwise I'd be in big trouble today. So that was what motivated me. So I thought, while it was happening, here were all these characters, and they all had like weird things about them. And also my own life was full of all these mistakes and blunders. And even while we were solving the ribosome, we were making horrible mistakes, And I thought, it would make a great story. So initially, this is interesting. Initially, I wrote the book, and I started the first two chapters, like a long history of molecular biology ending in the ribosome. And I showed it to my editor, and he said, "What do you think you're doing?" I said, "Well, I'm writing my book." He says, "Look, this is not a textbook, okay?" He said, "You're going to bore people in chapter 2, and then they're not going to read your book again. They're just going to put it down." And he said, "Just get on with your story and dribble the science in as you go, because people have to be invested in the book." So what I did was, that's exactly what I did. I started off with an episode that would grab somebody's attention. It was when Ara Yonat visited Yale when I was a postdoc. And then I start off with my story, and then I dribble in all of the science as I go, as I need it. And so it gives it a little narrative structure. And if you find the science a little hard going, you still have the narrative to hang on to, so you can just sort of skim through it and still enjoy the book as a story. So that was the goal.
That's great. Thank you. So we are going to move on to the book signing next. I think that's our next engagement here. And so I just wanted to thank you for coming and sharing your story with us. I had the good luck of going also to Venki's science talk earlier this afternoon. It was beautiful. So let's just thank Venki. You're really an inspiration.
Thank you.
Thank you so much.
And thank you so much for taking the time to spend with me.
Oh yeah, it's my honor.